So without further ado, I'm pleased to introduce our morning moderators, Dr. Erica Fink and Cliff Calloway from Critical Care Medicine and Emergency Medicine, respectively. Just one Zoom housekeeping note, please remember to post your questions in the Q&A, not the chat. We would like to be able to use the chat for administrative purposes. With the questions in the Q&A, the moderators can then accumulate them and ask them at the end of each presentation. And remember to keep your mic muted so that any background noise doesn't disturb the presentation if you happen to have a live mic. So enjoy the session, and I will unshare and turn the program over to Drs. Fink and Calloway.
Hey, thanks, Pat. This is Cliff Calloway. We'll introduce ourselves. I'm a professor in emergency medicine and one of the co-directors in the Safar Center. Erica?
Hi, sorry, I couldn't open my video there. I'm Associate Professor of Pediatric Critical Care Medicine, and really excited to hear the speakers this morning. Thank you.
Pat laid out the ground rules. We have about a half an hour for each speaker, and we are hoping to hear a presentation and then have time at the end of that half hour for questions and answers. One of the advantages of doing this virtually, the Q&A is open through the whole talk. So if you are thinking of a question, you don't have to sit on your hands like usually. Go ahead and put it in the Q&A. Again, we're going to save the chat for technical problems. So try to put your questions in the Q&A. If it goes in the chat, we'll try to catch it. It's not a disaster, but we'll try to use those two channels. I'm pleased to introduce our first speaker, Dr. Mark Sherdak from the University of Pittsburgh Drug Discovery Institute. So great to have somebody from our home institution to kick us off. Mark is associate professor in computational and systems biology. He has developed over his career many efforts in drug discovery. And I think the theme has been developing more effective drug discovery strategies and techniques, particularly strategies for high throughput screening using integrated function-based analyses of drug candidates. Over the last 9 years, he's been developing quantitative systems pharmacology, an approach to drug discovery that integrates clinical, computational, and experimental analyses. Again, that's going to be a theme, I think, of our morning session, and is in particular looking for effective approaches for disease management in neurodegenerative diseases like Huntington's, Alzheimer's, and TBI. So Mark, we'll turn it over to you, and please put your questions in the Q&A, and we'll catch them at the end. Mark.
Well, thank you, Cliff, for the introduction, and thank you, Pat, for the invitation to participate in this symposium. So today I'm going to talk about the quantitative systems pharmacology approach to target trauma-related neurodegeneration. So I'm going to go through a brief introduction to the TBI problem and our program goals, and then outline the quantitative systems pharmacology, or QSP, platform for understanding disease mechanisms and identifying therapeutic strategies, and then describe the experimental and computational workflows that we've been employing for addressing TBI, and then summarize what we've learned so far and where we're going. So, as you probably know better than I, traumatic brain injury is a major health issue. There are 2.8 million traumatic brain injuries a year in the US alone. And while the majority of them are treated and released from the ER, a fraction, 15% or so, are diagnosed with mild TBI and have long-term problems. The direct and indirect costs of TBI are staggering, $48 billion annually. And for those patients living with severe TBI, it can cost them $4 million over their lifetime. Now, as you know, TBI pathology is complex. There's the primary injury and then secondary injuries that lead to neurological deficits. And the secondary injury includes the molecular, chemical, and inflammatory cascades that are responsible for further cerebral damage. And currently, medical interventions for TBI are primarily symptomatic, efforts to reduce intracranial pressure, efforts to prophylactically address seizures, and in some cases, surgical interventions. So towards developing more effective therapies, we need to understand, I think, what those secondary insults are. After the primary mechanical injury in TBI, a series of secondary insults occur from seconds to minutes to days later. And these processes lead to impaired blood flow, metabolic imbalances, tissue damage, cytotoxic edema, inflammation, and ultimately cell death. So effective therapeutic strategies would include managing the critical mechanisms involved in the evolution of the secondary injury to prevent the initiation of secondary injury cascades, also to augment any repair mechanisms that occur due or to counter the damage. And while these processes are well accepted as being involved in the secondary insults, the details of the molecular targets in these processes are not really clearly defined in the context of TBI. So the goals of the quantitative systems pharmacology programming, the ultimate goal is to develop effective therapeutic strategies to mitigate the development of the secondary injuries in trauma-related neurodegeneration. And this will be accomplished by employing an integrated QSP approach to understand the mechanisms of trauma-related neurodegeneration. And that involves computationally inferring pathways and networks that are involved in the in the damaged or disease progression based on the RNA-seq profiles of injured versus non-injured neurons, since this represents the cellular response. And then using that information to identify known drugs or predict drugs that interact with these networks for experimental validation. And like in many complex diseases, multiple mechanisms are often involved that need to be addressed, and combinations of drugs that address or affect the different mechanisms of the disease are required to have a more effective overall therapeutic efficacy. And then finally, advancing the TBI protective compounds that we identify to the clinic. So what is TBI? I'm sorry, what is QSP? Too many acronyms. Quantitative systems pharmacology, or QSP, is functionally defined by us as determining the mechanisms of disease progression and mechanisms of action of drugs on multi-scale systems through the iterative and integrated computational and experimental methods to optimize the development of therapeutic strategies. So how does it work? QSP begins with a focus on the patients, from whom a wide range of patient-specific clinical and omic data are acquired. These data represent a distribution of patient genotypes and disease phenotypes. And these data are input for computational analysis to infer pathways of disease progression that contain a collection of potential therapeutic targets. And these form the starting point for understanding mechanisms of disease progression and what pathways to modulate in order to correct the disease phenotype. Drugs and probe compounds are identified for these targets using machine learning tools that predict the drug interactions based on information analyzed in databases such as DrugBank and Stitch. And these two probes are then tools to learn about the mechanisms involved in the generation of the disease phenotype. Phenotypic models are developed that reflect the patient-derived information, including the heterogeneity of the responses among the patients. The predicted compounds are tested in these clinically relevant phenotypic models to probe the dependence of the canonical target on the disease phenotype. RNAi knockdown and mutant libraries, as well as focused compound libraries, are also tools that can be used at this point to aid in uncovering the mechanisms, as well as a starting point for identifying novel leads. The effect of the probe compounds on disease phenotypes are analyzed on a cell-by-cell basis to characterize the subpopulations, including the presence and extent of heterogeneity of response to the treatment. Multiple functional parameters are measured, and the data yields a systems-level response involving potentially involving multiple pathways that need to be addressed to reduce or moderate the disease phenotype. Hits from this phenotypic profiling demonstrating the ability to reverse the phenotype can then serve as starting points for drug repurposing when the predicted drugs show results, or a starting point for the development of novel therapeutics from compounds from the focus libraries, for example. Medicinal chemistry efforts can help to optimize the predicted drug hits, both for repurposing drugs for new indications as well as for optimizing novel compounds. The lead compounds are then tested in vivo disease models to validate the efficacy and safety in a multi-scale system, which is a more complex system relative to the in vitro systems. Lead compounds also undergo target identification using a combination of approaches such as NMR to validate their activity on the canonical targets that they were derived from, or to identify novel targets that they may be interacting with. Successful targets that have been identified at this stage can be directed to a more traditional target-centric drug discovery development, using that target in other structure-activity relationship studies for the development of compounds. Keeping in mind that anything that comes out of those studies will also need to be able to reverse the phenotype in the more phenotypic assays. Computational models of disease physiology and progression are created by integrating the information that's generated in all of the steps before this, as well as combining with validated target knowledge that is in the literature. Now, these computational models or these initial models are incomplete models, but iterative steps around the QSP cycle allows the refinement of the experimental models, the identification of additional probe compounds, and further refinement of the computational models. An iterative process of experimentation and computational modeling converges on a comprehensive characterization of disease progression with increasing accuracy and identifies disease-specific emergent properties, including the identification of novel therapeutic strategies, pharmacodynamic biomarkers, and prognostic and predictive biomarkers. And the knowledge gained from these iterative steps guides clinical trial design, with which the clinical trial itself becomes now an integral part of the module of QSP cycle, which generates additional patient data to further refine the understanding of the pathophysiology and the design of the next generation therapies. So in applying the QSP approach fundamentally to TBI, our workflow starts with understanding the transcriptomic changes that occur with mechanical injury to neurons. We use a well-established stretch model, a stretch injury model, to measure the gene expression changes in primary rat cultures 24 hours after the injury. From the differentially expressed gene profiles, we computationally infer the pathways that were modulated in response to the injury and identify molecular targets in those pathways, and then finally identify probe drugs and compounds that are predicted to modulate those targets to be able to validate those pathways in the disease phenotype. We evaluated two levels of stretch injury, a low level of stretch injury, which is imparted by a 54% stretch level in the experiment, and a higher level of injury, a 75% stretch in the experiment. The goal was not to induce severe injury and kill all of the cells, but to induce a mild form of injury so that we can assess the molecular mechanisms that are modulated. We saw the expression of over 3,000 genes modulated to some degree after both injury levels. Overall, the gene expression profiles were similar between the two injury levels. Generally, the degree of modulation seen in the low injury condition was less than that seen in the high level. As you can see, the green bars here on the graph for the low injury represent the levels of change of genes, and the red bars represent the levels of change in the high stretch injury model. 95% of the genes that were upregulated by low stretch were also upregulated by high stretch. And similarly, 94% of the genes that were downregulated by low stretch were also downregulated by the high stretch. And so we have this a common set of genes that are following the same patterns after both low and high injury levels. And these genes are considered to be the common genes associated with the injury response. The minor subpopulation of genes that differed in their direction, expression between the two levels of injury is interesting, and is something that we want to be following up later on. So using the gene expression patterns identified, 176 pathways were inferred to be modulated by the stretch injury. 137 of those inferred pathways were consistently regulated by both injury groups. 75 pathways were predicted to be activated in both the high and the low stretch condition, and 62 pathways were predicted to be inhibited in both conditions. Now, all of the genes in those pathways are considered potential targets for modulation and therapeutic intervention. And so while we take a gene, such as gene 1 here, that will code for one protein, and that protein is what was used to identify what the pathways involved, that gene represents a therapeutic target. However, to expand the relevant target set, we considered all of the targets in that pathway as being important and potential therapeutic targets downstream. And so we've included those in our analysis as well. Now, compounds for all of these targets were identified through interactions listed in the STITCH database. And then signaling pathway impact analysis was used to predict the compound effects on the pathways, either enhancing the pathways or inhibiting the pathways. From this analysis, 82 pathways were prioritized and 331 probe compounds were identified. Now, in this chart, we have listed on the right side on each one of the rows are the 82 pathways that were inferred. And on the bottom, we have the 331 compounds that were identified. Now, the top part of this heat map, represented here by the blue bars, are those pathways that were inferred to be downregulated after the stretch injury. And the bottom part here represents those pathways that were upregulated after stretch. Now, we've identified both enhancers and inhibitors of these pathways. So we have a set of compounds identified that would enhance the downregulation of the pathways or enhance the upregulation of these pathways, as well as inhibitors of upregulated pathway and inhibitors of the downregulated pathways. At this point, we don't know if the upregulated or downregulated pathways are the causal effects of the secondary injury or if they're part of a repair response. And we also don't know if we need to enhance the activity or reverse the activity, at least at this point. And so for a number of the pathways, we have identified both inhibitors and enhancers, which will help elucidate which way we need to be driving the affected pathways to limit the neuronal damage due to the stretch injury. The next step was to validate the activity of these predicted drugs and learn which pathways are important in the biological response. The compounds were tested using the same stretch injury model under the same conditions that we used for the initial RNA-seq analysis, except we only looked at the high stretch level condition. We used an image-based high-content assay to monitor the loss of neurofilament light chain from the cells as an indicator of axonal damage and the ability of the probe compounds to protect against it. Now, as you know, NFL is a neuronal cytoplasmic protein. It is used clinically and measured in the CSF and the plasma to assess the degree of axonal damage in a variety of neurological disorders. And so it made a good biomarker in our case to assess the clinically relevant damage in our model. And the high content approach was used because this approach allows us to measure multiple endpoints, such as morphological features, as well as other pathway markers to evaluate the effect of compounds on their canonical targets and downstream. So here's an example of the control for the stretch injury. On the left, we have our vehicle control with 0% stretch. So these will represent uninjured neurons, stained for the neurofilament light chain. As you can see, we have fairly intense staining in these cellular structures. After 75% stretch, you see that there's a diminution of the staining of the NFL. And we've developed algorithms to be able to quantify the levels of the NFL in these cellular structures. And we found that overall, with this 75% stretch injury, there's about a 40% drop in the NFL levels in the cells. And so we have a fairly good window with which to assess the effects of the compound and their ability to reverse or protect the axonal damage. So we are testing our predicted compounds, and this is just showing some data from one of the early experiments where we've tested a number of compounds in the same stretch model. Here we've normalized the NFL intensity to both the control or the uninjured control or 0% stretch, as well as the 75% stretch. And as you can see that we have there are various effects of the compounds in their ability to protect from the loss of NFL due to the stretch injury. We have compounds that seem to protect, seem to do nothing, and some that may be more detrimental. The dark blue lines are those which are significantly different from the stretch control, the 75% stretch control. And so from this analysis, it appears that several different pathways may be involved in the protective response. Pathways including the glutamatergic pathway, cholinergic synapse pathway, the renin-angiotensin pathway, IL-17 signaling pathway, renal cell carcinoma, or PI3-AKT signaling pathways. Compounds that are associated with modulation of these pathways are showing what appears to be a protective effect in this model. Now, this was one of the early studies. And so what are we doing now is that we are confirming those activities, characterizing them further in terms of the potency of these compounds and the efficacy in concentration response assays. In the initial experiments, we chose to screen at a single concentration in order to get a fair number of replicates for each compound, as well as getting a larger number of compounds tested, since the stretch assay is a relatively low throughput assay. We are also looking at characterizing the heterogeneity of the response heterogeneity of the different the cell populations in their response to the treatments. After confirming the activity of these compounds, we will be determining the engagement of their canonical targets. Again, it's important to know that they are working on the targets because the whole analysis is predicated on the activity of these compounds on their canonical targets. Yet we know that the compounds can interact with multiple targets, which is one of the reasons why we have side effects or off-target effects in the clinic. So it's important to establish whether or not the canonical target mechanism is acting, or if there's a novel mechanism that's being engaged. Once we've established with the canonical targets being engaged, we will be exploring the dependence of those targets on the phenotypic response. And obviously, we are continuing to work through the list of compounds because additional compounds will be targeting different pathways, and this will expand the scope of the pathways involved potentially in the response, and this will help refine the computational models as well as direct the design of the therapeutic strategies. On the computational side, using the information gained from the above experiments, we can start predicting combinations of active compounds to be able to enhance the activity. We will be looking at the convergence of the pathways from the transcriptomic profiling, asking the question is that, do the pathways converge on critical functions in the cells that will guide the development of therapeutic strategy? And we'll also be looking at how to see how the inferred pathways relate to the clinical biomarkers used now, and to inform or predict novel biomarkers for axonal damage that may be useful down the road. So in summary, we have applied the QSP approach to understand the mechanisms involved in axonal damage after a TBI injury, looking at the transcriptomic response in the injury to begin with. We have inferred a number of pathways that may be involved in the cellular response, along with a number of probe compounds to tease out those pathways critically involved in the response. This is still a work in progress, and iterations of computational and experimental analyses will enable us to develop and refine both the experimental models as well as computational models to cellular response. And ultimately, this will lead to a comprehensive characterization of disease progression and identify the disease-specific emergent properties. And the knowledge gained from this approach will guide the design of effective therapeutic strategies and clinical trials. Go the other way. So at this point, I'd like to acknowledge the many people who are involved in this project, many people at the University of Drug Discovery Institute, people involved at the Pharmacology and Chemical Biology Department, the Safar Center. Pat Kahanek is very helpful in guiding what, the design. Travis Jackson at the University of South Florida. And finally, want to acknowledge the funding for the PA Cures Grant with David Okonkwo as the PI. And at that point, I'll be happy to answer questions.
Mark, fantastic. Thank you so much. You don't benefit from the applause we have, so we'll give you virtual applause. There are a couple of questions in the Q&A, and if folks have questions, go ahead and post them in the Q&A. I'll start. Dr. Yaffe asks, are there targets and therapeutic approaches that have also got application for post-traumatic stress disorder? Is that an entity that the Drug Discovery Institute has looked into?
So a target that we've identified through the current work, or
well, yeah.
So I think
yeah, Doctor. Yeah, is PTSD on your radar? Is something to look for drug candidates?
At the current moment, no, it is not. But it's certainly something that we could entertain.
Another question, Dr. Zisman, have therapies that have previously been found to be beneficial in preclinical models, like levetiracetam or glyburide, how have they performed in NFL screening models? So do you have positive controls, I think?
So actually, yes, we have tested those, and currently we have not seen a robust response with those compounds in this model.
Yeah, it's interesting. I wondered as you're describing this, I mean, many things can happen, and if you have a readout like NFL quantity, how do you look for other effects that might be important for the recovery of the patient? Other pathways that simultaneously may be involved.
In terms of the activation of the canonical targets, for example.
Yeah, for example.
Things you can do using a high content approach is obviously we can label multiple targets within a cell. And if, for example, you're looking at inhibition of a kinase pathway, you can look at downstream phosphorylation of the targets or inhibition to assess whether or not that kinase has been affected by the treatment.
So is this part of the process where you narrow down candidates? So, for example, if you come to me with 90 candidates, it will take the rest of somebody's career to test them all in a large animal model. So, how do we decide which one is the next one?
So, obviously what we want to do is make sure or identify what the targets are and that the compounds are engaging those targets to better understand the pathways that are involved. At this point, we are working with a rat cell model, and obviously we want to be able to cure the disease in humans. So part of the QSP approach is the development of clinically relevant phenotypic models, and that would be including models such as microphysiology models that would reflect the disease phenotype. And so we would then go into testing those compounds in those models, which can then, the data can be used to prioritize the compounds to move forward through animal models and then into the clinic.
Other questions? Let me ask one to wrap up. Oh, Pat has a question. Go ahead.
Good.
I was wondering if there would be some thought into the timing. You waited till 24 hours. Do you think you might get very different signals if you went to like the immediate response versus a delayed response?
So yes, and then that has been something that we have discussed, whether or not we should be looking at a more proximal time point. And one of the things I think we would like to do, something we haven't done yet, is to be able to do a time course of the transcriptomic changes. So yes, I think that will influence the direction. It's just something we haven't done yet.
Hi, Dr. Sherdak.
Yes.
I'm curious if this model allows for any combined testing of interventions.
In terms of combining compounds?
Yeah, combining two different treatments together.
Yes. Yes, and that is certainly something we want to do after we've confirmed the activity and confirmed the targets that are being involved in the reversal of this phenotype. We can then computationally predict, well, these are the ones that you probably want to combine to have a more efficacious effect. And we can test them in this.
Great. I don't see any more questions. Thank you so very much, Dr. Sherdak, for your talk. And I look forward to hearing more about your work. And we'll move on to our next speaker. I'll introduce Dr. Manuela Keller. I'll get her up. It's my pleasure to introduce to you Dr. Keller. She's an associate professor of intensive care at the University of Zurich in Switzerland, and she's chief of neurointensive care there. She has an extensive training history and including fellowships with in leadership and in informatics. And it looks like she's combined all of her training into some fantastic research focused on new methods to estimate cerebral hemodynamics and oxygenation using optical spectroscopy and clinical research informatics. In addition to being extremely well funded, she has many patents for her inventions and has a spin-off company, which sounds like it's award-winning. And I'm really excited to hear about her work today. Thank you.
So thank you very much. I just tried to share my screen. Dear Dr. Kokenek, dear Dr. Callaway, Dr. Fink, thank you very much for inviting me. It's a great pleasure to have this lecture in this honorable symposium. My disclosures. So if we want to take a look into the future, it's worth looking back into the history of neuromonitoring. Brain ventricles and their relationship to the pathology of increased ICP has been studied for several centuries. However, it was not until the hypothesis by Harvey Cushing that the role of CSF in ICP was really understood. Harvey Cushing, inventor of the anesthesia protocol. He monitored the heartbeat with one of the first stethoscopes, traveled to Europe, to Pavia, Italy, and brought home the newly invented sphingomanometer from Rivarocci. Professor Cushing realized very early the importance of circulation and oxygenation in order to avoid stress focused on ICP. In 1927, Edson and Lilly, a neurosurgeon and ophthalmologist, published a landmark paper relating ICP and intraocular tension, focusing on manometry. Henceforth, a manometer has been attached to the head of the bed. In 1960, Niels Lundberg from Sweden transferred ICP monitoring in mostly brain tumor patients. The work provided minute-by-minute analysis of ICP and waveforms that firmly established the feasibility of prolonged drainage. ICP waveforms are named for Lundberg's contribution to the correlation between heartbeat, respiration, and ICP. As you know, in 1995, the Brain Trauma Foundation guidelines provided some consensus with recommendations for ICP monitoring in any trauma patient with Glasgow Coma scores below 9. Most of you know the Best Trip trial, concluding that ICP-guided care was not superior to care based on imaging and clinical examination alone, contrary to published guidelines. The study has significant limitations. However, ICP elevations may be an insensitive and late indicator of secondary brain injury, and monitoring and treating other physiological variables may enhance patient care. As mentioned by Peter Leroux in this subsequent review, multimodal monitoring should be more widely adapted to better understand the complex pathophysiology after traumatic brain injury. So BOOST 2 up to date is the only randomized controlled trial examining the use of brain tissue oxygen monitoring in clinical practice and demonstrated the feasibility and safety of PbTO2 monitoring in TBI patients. Currently, 3 ongoing multicenter randomized controlled trials studying the use of PbTO2 monitoring in severe TBI are running. In highly specialized centers today, we perform multimodal monitoring, applying ICP PBTO2 probes, near-infrared spectroscopy. So I think if we want to take a look into the future, it's worth looking back into the history of neuromonitoring. So brain ventricles and their relationship to the pathology of increased ICP has been studied for several centuries. However, it was not until the hypothesis by Harvey Cushing that the role of CSF in ICP was really understood. Harvey Cushing, he is very well known as a pioneer in neurosurgery, but only few people know that he's the inventor of the anesthesia protocol. He monitored the heartbeat with one of the first stethoscope, traveled to Italy, to Pavia, and brought home the newly invented sphingomanometer from Riva-Rocci. Professor Cushing realized the importance of circulation, oxygenation to avoid secondary brain image damage very early. So traditionally, neuromonitoring has focused on ICP. In 1927, Edson and Lilly, a neurosurgeon and ophthalmologist, published a landmark paper relating ICP and intraocular tension focusing on manometry. Henceforth, a manometer has been attached to the head of the bed. In 1960, Niels Lundberg, he transferred the ICP monitoring into clinical practice and published an extensive analysis of the use of ICP monitoring in mostly brain tumor patients. The work provided minute-by-minute analysis of ICP waveforms, and he established really the feasibility of prolonged CSF drainage. ICP waveforms are named for Lundberg's contribution to the correlation between heartbeat, respiration, and ICP. As you know, in 1995, the Brain Trauma Foundation guidelines provided some consensus with recommendations for ICP monitoring in any TBI patients with a GCS score below 9. Most of you know the BEST TRIP trial, concluding that ICP guided care was not superior to care based on imaging and clinical examination alone, contrary to the published guidelines. The study has significant limitations. However, ICP elevations may be an insensitive and late indicator of secondary brain injury, and monitoring and treating other physiological variables may enhance patient care. As mentioned by Peter Leroux in a subsequent review, multimodal monitoring should be more widely adapted to better understand this complex pathophysiology after TBI. BOOST 2, up to date, is the only randomized controlled trial examining the use of brain tissue oxygen monitoring in clinical practice and demonstrated the feasibility and safety of PBTO2 monitoring in TBI patients. Currently, there are 3 ongoing multi-center randomized controlled trials studying the use of PBTO2 monitoring in severe TBI actually. So today in highly specialized centers, we perform multimodal monitoring, applying ICP, PBTO2 probes, near-infrared spectroscopy, and cerebral microdialysis, providing multiple data in real time at the bedside. So where are we going to? What are our clinical needs? I think most of all, we have a very high need for data integration. Data integration is essential for the following reasons. Only the presentation of complex data in a comprehensive way allows for better understanding of pathophysiology. For instance, in this case, you can see the relationship between the arterial blood pressure, ICP and CPP, seems to be less important than, for example, the cardiac output for the occurrence of cerebral ischemia, here reflected as an elevated lactate pyruvate ratio measured with cerebral microdialysis. The influence of a single parameter as a trigger for cerebral ischemia can only be detected if data are integrated and visualized all together. Second, data integration is needed to halt vicious cycles of physiologic deterioration, and even more important, to develop algorithms to predict imminent complications, which allow for preventive measures to avoid them. And finally, data integration will allow to adapt treatment to the very individual needs of every single patient. Conventional evidence-based treatment is based on a treatment failure approach. We apply treatment A, observe its effect, if not helpful, treatment B, and so on. Treatment B may be too late if cerebral ischemia occurs. Precision medicine allows a proactive approach, not only the disease itself, but we take into account multiple parameters for a personalized treatment. During the ICU stay, as we already heard before, even much more data for multimodal monitoring can be taken into account, which allows for precision medicine. With this demand for data integration in mind, in 2014, we started the project ICU Cockpit as an integrated IT platform for patient monitoring and therapy support in neurocritical care. Data are collected systematically behind a thick wall for data encryption, which is the firewall of the hospital. We have researchers from the university, ETH, and IBM Research in Zurich developing algorithms for early systems and therapy support. The models developed are validated and finally transferred into software, and the results are visualized for the medical staff at the bedside in real time. I want this lecture to explain to you the overall IT infrastructure we have set up until today, only very shortly. The data are collected from different medical devices. So actually, up to now, we have developed several algorithms as for signal artifact elimination, automatic seizure detection, based on video and on EEG monitoring. We try to understand pathophysiological relationships between different parameters as ICP, blood pressure, and central venous pressure. And finally, we try to predict DCI, delayed cerebral ischemia, and brain tissue hypoxia in patients with aneurysmal subarachnoid hemorrhage. As an example, allow me to show some results about DCI prediction from the Columbia Group and our own preliminary data. So traditionally, grading scales are performed in SH patients on just static assessments of the clinical condition, certain risk factors, radiological findings on CT scans at admission, and are associated with different odds ratios of outcome. So the Columbia groups, in addition, explored time series physiological data as heart rate, blood pressure values, respiratory rate, and oxygen saturation for the early prediction of DCI. An unsupervised approach applying random kernels was used to extract the features from physiologic time series, and combining baseline characteristics and features from physiologic time series improved the prediction to an AOC of up to 0.77. So published most recently, the same group took two very important next steps. First, they anchored patients' data to a median DCI onset to day 8, which allowed to capture the temporal dynamics leading to DCI onset. Hourly risk scores to the developed DCI could be calculated for each single patient, and second, they validated their models on external data sets, independent cohorts in Houston and Aachen. In our own research group, we investigated biomarkers for the occurrence of DCI, more than 250 changing variables, parameters from the medical history, lab values, values from blood gas analysis, and so on, were processed in a random forest model. You can see here the time course of the 10 most variables for a time window from 24 hours before the occurrence of DCI to 3 days afterwards. The chronological progression of the biomarkers show that, for example, interleukin-6 in red and CRP in blue are obviously important as markers of the cerebral inflammatory response. So this was preliminary work to identify the 10 most important parameters at baseline as gradings and risk factors, defining inputs for a so-called static model, and to select the 10 most important lab values as inputs for a dynamic model, both based on random forests. Finally, we combined both models to a so-called voting model. We anchored the data to day 7, the most frequent day of DCI occurrence in our population, which allowed as for a daily risk assessment in every single patient. The model output is interpreted as the probability calculated finally based on the number of trees in the random forest going into a certain direction of plus or minus occurrence of DCI. So in the ROC analysis, the voting model combining the static and the dynamic model performed best with an AOC of 0.74. However, ROC analysis does not really reflect the performance of a model in a single patient over the course of the disease. Therefore, that's why we performed a so-called leave one out algorithm simulation, taking the following steps. First, a single patient is excluded from the training set. As a second step, static and dynamic models are trained on anchored data. And finally, risk assessments for the left out patient over the course of its entire stay on the ICU are performed. The procedure is repeated for past and currently actual patients in a silent validation mode on the live data. Let me show one example of the model simulation. At the left side, the results from the static model is reflected in a so-called waterfall plot as a blue arrow characteristics reducing the risk for DCI, as for example, the lower Hampton Hess grade are reflected in red. Parameters increasing the risk as the high BNI grade of 5 or the presence of hydrocephalus are given. Only taking the static model into account, the risk to develop DCI for this patient is 0.687. If we just take the static model into account, you can see the probability of DCI remains the same over all the time. You can see this reflected in the blue line in the upper right graph. So the blue line stays constant during the entire period. However, as we know, this does not really reflect the reality. However, if we take the dynamic model based on the lab values into account, the situation changes. In the lower graph, you can see a heat map for the lab values over the time course. In the late phase, the importance of the inflammatory biomarkers increase. In the upper graph, you can see in orange the probability for the dynamic model alone, and finally in green, the probability for the combined, the voting model. The voting probability increases on day 11, and indeed, a new neurological deficit occurred in this patient on day 12, combined with a perfusion deficit in CT scan on day 13. The patient was treated with PTA, balloon angioplasty, and intra-arterial nimodipine twice. Let me come to conclusions. I think data integration, data mining provides a very powerful spectrum of predictive tools, prevention and early interventions, as well as for individualized treatment, especially based on neuromonitoring. However, only a very few algorithms developed have been transferred to clinical practice up to now. As in other technical innovations, we can talk about the AI chasm. So what are the obstacles? First, I think in most studies, data are collected very selectively. Data quality, provenance have to be carefully checked, which otherwise might lead to a algorithmic bias. We need external validation of algorithms before models can be generalized to other populations and settings. Treatment standards change over time, so algorithms have to be retrained. For clinical studies with medical device software, we know we have very high regulatory hurdles, which do not really fit to these big data issues. Clinical staff will only trust, as already mentioned, algorithms results if they are really transparent. So I think explainability of AI-based models results became to a very important field in medical informatics. And finally, accuracy do not really reflect necessarily clinical efficacy. So I think validation on live data streams and in the ICU environment are very important. Finally, let me refer to an article recently published in Nature Medicine, providing a very far-sighted view and guidelines for accelerating the translation of machine learning-based interventions in healthcare. The authors propose a roadmap involving stakeholders from the very beginning, from the medical domain, very early, for the development of useful solutions over ethical implications, clarifying responsibilities up to the medical device market. And with this view from our hospital over the university center, the city, and the Lake of Zurich, I thank all our collaborators and you for your patience. Thank you.
Thank you so much, Dr. Keller, for that great talk. I'm really glad that we're able to get it in its entirety, this great body of work. I know that we are looking to get questions in the Q&A. And while we're waiting for a question from the audience, my question to you is, now that you've established a model that appears to have decent accuracy in predicting the onset of ischemia in this particular condition, what do you think are the next steps? What is missing from the model that you haven't been able to capture that could help explain the incidence of ischemia to predict it?
I think as a very next step, we have to implement the high resolution data streams. So for example, currently we are developing algorithms to calculate the heart rate variability, blood pressure variability, and to implement analytics from those high time resolved data streams into our predictive algorithms.
Thank you so much. And we have a question from Dr. Barney Schofield in the UK, PICU clinician. Excellent talk, thank you. Can I ask how we can adapt the AI approach to the much smaller numbers of patients and therefore outcome events that we have in the pediatric neurointensive care world? Or do we need to stay away from AI? Thank you, Barney.
No, I think you don't really don't have to stay away from AI. Even in pediatric or neonatal ICUs, it is very important, for example, for artifact elimination to develop smart alarms in this patient population might be very helpful. I think if the patient numbers are too small, it is really essential to collaborate with other centers. And finally, I think not even in adults ICU care, it is very important that we share our data, that we have the option really to validate our algorithms on external data sets to generalize the predictive algorithms to other population groups. So collaboration in this field, I think, is very essential. But to have this potential for collaboration, it is there is a very high demand for standards. So for example, to have databases which are really comparable, and I think this is a call to the industries that they really should standardize, for example, the data format, the type of data which are really exported from the devices. There are no standards as, for example, in radiology, which has the DICOM format. We really need some established standards in that field for multi-data collecting databases from multiple centers.
Thank you very much. I think in the pediatric world, we are definitely having to use our networks and partners, various partnerships and collaborations to answer the bigger questions, thanks to our smaller sample sizes and less severe events. Any other questions, Dr. Keller? I also appreciate the call for being able to harmonize data elements and as you did, making it very difficult to compare data across centers, let alone countries. Okay. Pat, you have a question?
Yeah, Dr. Keller, thanks for hanging in there through the technical challenges. Delayed cerebral ischemia after subarachnoid hemorrhage is kind of a unique entity in neurocritical care. And I'm wondering if this approach might not, your thoughts about the utility of it literally in the first 15 minutes or 30 minutes, or at most say 1 hour of providing us with so much more insight on what the directionality of a patient will be. Do you see its use in this kind of immediate application?
So currently we have a dashboard on our ICU, which really visualizes the prediction for every single patient. And to be honest, it's not always really plausible what is shown by this algorithm. But I think especially in the development of DCI in SH patients, we have so multifactorial inputs that with our conventional approach, to estimate a certain risk for patients, we are really overwhelmed. And I think it is the disease, subarachnoid hemorrhage, with the development of DCI and vasospasm is a very important patient group really to apply those predictive algorithms.
Dr. Callaway?
Yeah, just a quick question. Thanks for an excellent talk. I was really interested in your comment about the gap in or the chasm in AI deployment. And, you know, I perceive that there's great suspicion from clinicians when something is proprietary and that we don't know what goes into an algorithm. Yet, you know, companies are going to provide us with the technology to bring algorithms to the bedside. And, you know, is there anything that you've envisioned that would help make clinicians more trusting of companies and the entities that will actually make algorithms available for patient benefit, or, you know, other education that we need to be doing all through the medical profession to help make that chasm less of a barrier?
I think it is very important to involve the medical staff, even the nurses, at the very beginning. So if you really visualize results from algorithms at the bedside, not only the explainability for the medical staff to trust, but the usability, the user friendliness is very important. And we have involved, for example, psychologists in our research group too. Psychologists can study this machine-human relationship, which has a very important which is a very important aspect for user acceptance in daily clinical practice. So it needs a very lot of education of medical staff in clinical practice, really to train them to use these models in the daily practice, that they do not just realize it's another computer interface. We have to apply this in the practice. It really has to be helpful, supportive for them in practice. And the other thing I think would be very important, at least in Europe, we have huge regulatory hurdles. So for example, actually now visualizing the results from our algorithms, the type of study is now an interventional clinical study. So for example, I have to report all the serious adverse events which are not really related to the software anyway. So I really had to employ a full-time regulatory person to realize this clinical study now, interventional trial, investigating algorithms in daily clinical practice to overcome this workload from the regulatory point of view. So I think to have new types of study regulations which are really adapted to this type of research would be very helpful. But I think the FDA is much more far in that direction now going than we have our regulations in Europe.
Fantastic discussion. Thank you very much, Dr. Keller.
Fantastic. Well, thanks for being flexible. Let me then go ahead and introduce Jonathan Elmer, who is one of my colleagues in emergency medicine. Jonathan is also a neurocritical care physician and is one of the smartest people that I know. He has been doing a great deal of work on intensive neurophysiological monitoring in patients, particularly in cardiac arrest patients, and using some large data approaches to understand the physiology of that situation and how we might better intervene with people's brains as they recover from cardiac arrest. So thanks for stepping in off schedule. And Jonathan, we'll turn it over to you.
Thanks so much, Cliff, for the kind introduction, and thanks to the entire organizing committee for the privilege of presenting to you all today. I thought I would talk a little bit about using continuous EEG after cardiac arrest with a bit of a sort of hybrid approach, thinking both from the perspective of the bedside clinician, but also from the perspective of the research scientist. We'll start briefly by just talking about the why and what of EEG, which many in the audience may know already. I'll touch on some of the challenges and problems working with continuous EEG, both clinically and particularly from a research perspective, and then introduce a couple proposed solutions briefly. These are my disclosures. I think none particularly relevant for this talk, except for some grant funding related to the work I'll be presenting here today. So I admit to being biased. I'm a bit of an EEG enthusiast, but I think it merits brief comment on why I think that EEG is such a valuable signal to guide critical care and to guide prognostication. This is an EEG for those who have not seen one before. This is about 8 seconds of continuous EEG recording. We record EEG by gluing little metal electrodes to the patient's scalp and then measuring the voltage difference between various combinations of electrodes. And what you end up with is these sort of squiggles that go by continuously. And again, this is about 8 seconds of data there. And what makes EEG very useful, but also potentially very challenging to work with, is the complexity of the inputs that generate this incredibly rich signal. So there are aspects of the integrity of cortex, the networks between cortex that generates the EEG and various other deep brain and other structures that affect the signal. There are signals that can be teased out related to the well-being of the cortex from a functional perspective, the cortical energetics, healing over time, seizures and excitotoxicity and other mediators of secondary brain injury, like brain tissue hypoxia or hypoperfusion. And then there's also these signals that come from potentially modifiable aspects of critical care, things like the temperature, pharmacotherapy, adequacy of the blood pressure to perfuse the cortex, hemoglobin, and other determinants of oxygen delivery. From the perspective of cardiac arrest in particular, that I'll focus on, I sort of bin these in my mind into stuff that is related to the primary anoxic brain injury, and thus may have some prognostic significance, much of which comes down to structural damage to the brain that occurs during the cardiac arrest itself. And then from the perspective of prognostication, other stuff that may be confounders, or from the perspective of clinical care, things that may be an indication of treatment responsiveness. And I'll talk about that a bit more as the talk goes on. From the research perspective in particular, we think about EEG either as being interpreted in a qualitative way. So you might look at this 8-second snippet and say that there's a continuous background activity here and maybe some spiky things that happen intermittently, or from a quantitative perspective. And given the theme of today's talk, I think I'll focus primarily on the quantitative analysis of EEG. So just as the whirlwind tour of what we mean when we say quantitative analysis, I've blown up a snippet of the EEG here and a single channel. This is about a second of data. And you can look at this second of data, the squiggles that go across the screen. And I think you can fairly quickly start to think about ways that you might describe this signal. So one thing you could do is you could measure from the baseline to the various peaks or inflection points where the signal sort of goes from upsloping to turning around. And you can measure the height of those peaks, and you can try to understand aspects of the amplitude. What is the average amplitude? What's the variability in that amplitude? You can use one of several techniques like a Fourier transform to try to understand what the component frequencies are, which may have clinical and prognostic significance. And then you can do a whole bunch of other stuff. And so this is a slide which sometimes I show like this, and sometimes I show the cartoon of Alice falling into Wonderland with all the stuff floating all around. It's truly a rabbit hole when you start to try to featurize these waveforms. So you can look at the amplitude and the variability of the amplitude and the variability of the variability over the amplitude. You can make probabilistic predictions, like how likely is the signal to represent something that an expert would think is a seizure. You can look at measures of synchronicity or cohesion among various anatomical regions of the brain to try to make conclusions about the cortical networks. You can look at the rhythmicity of the signal or correlations over time. This is both an incredible opportunity insofar as a robust mechanistic understanding of the signal can inform your analysis, but also a potential pitfall, because it's very easy to fall into this rabbit hole of increasingly complex and potentially less interpretable ways of decomposing the signal for analysis. So that is, I think, a prelude to some of the problems if you want to approach continuous EEG as a research, as a prognostic, or as a clinical tool. So there are truly infinite ways to quantify and extract features from this very, very rich signal. These are very, very highly multivariate time series for which there are imperfect analytical tools that exist now. They are very correlated, both over time and over space. So adjacent electrodes may have a lot of collinearity in the features that you can extract. And within a given signal over time, there's often strong temporal correlations. They're also incredibly large data. So our local EEG data, which is really only a few thousand patients and maybe 120,000 hours of data, has trillions and trillions and trillions of data points in it before you even start to try to derive features. That's just the raw waveform. And that poses some obvious computational challenges as you try to move dozens of terabytes from here to there, or execute even simple computational tasks. Importantly, in terms of statistical or ML modeling, there's also very informative missingness to this, like all observational data. So locally, for example, we monitor EEG continuously when folks hit the ICU after cardiac arrest until they get better and don't need it anymore, or until they get worse and don't need it anymore. And so you can't assume that the missing data are missing at random, which creates some potential analytical problems, although also opportunities. And then there's this very important concept, which I'll talk about more for the next few slides, of self-fulfilling prophecies. So what I mean by that is clinicians are looking at the EEG data that we now seek to analyze retrospectively or prospectively. And clinicians are sincere in their intents and would like to minimize futile, prolonged ICU care in patients who have irrecoverable brain injury after cardiac arrest. That's a noble goal. It is prudent in terms of directing limited resources towards those most likely to benefit. But it's also important to families to try to minimize the duration of what is potentially very uncomfortable, undignified, difficult to see critical care. So physicians want to limit care, or limit life-sustaining therapies, I should say, for patients who are unlikely to derive benefit. And they look at the EEG and they use this to inform their decision-making. And that means that often the EEG patterns that we see, either qualitatively or quantitatively, guide withdrawal of life-sustaining therapies, after which patients are likely to succumb to the sequelae of their critical illness. Those patients now, forevermore in our data set, are labeled as having a poor outcome. And so there's a strong association of EEG with outcome that's mediated by clinical decision-making, but not necessarily biological truth. So to sort of set this up, what I'm saying as sort of a stepwise scenario that I'll then build on, suppose that you believe me and EEG and other data are useful prognostically and they accrue over time. And so we watch them and we try to update our expected prognosis based on this continuous input of data. And suppose also that some patients recover after cardiac arrest and do well, and that some patients succumb to multi-system organ failure, brain death, or rearrest despite maximal medical support. But importantly, also suppose that many of our patients in the intensive care unit die after withdrawal of life-sustaining therapies based on their perceived neurologic prognosis, where that perception is shaped by the EEG. So it's these patients in particular in this last bin that we worry about from the context of self-fulfilling prophecies. And unfortunately, after cardiac arrest, this is a majority of deaths. So it's a common proximate cause of death. The conventional analytical approach is to take a bunch of predictors, including quantitative EEG features, but maybe also medications and vital signs and time invariant data like the patient's age or sex or initial rhythm, and to use some modeling tool to try to find associations between this set of features over here and the observed outcome. And so here, the observed outcome is either you recover or you die. The major flaw with that sort of an analytical approach is that you can't reconstruct the counterfactual scenario among patients who die, where you try to guess what their outcome could have been or what their recovery potential could have been in the absence of the clinical decision to withdraw life-sustaining therapies. And there are various analytical approaches to try to reconstruct that, but all of them are flawed. So I propose a simple alternative here that we have built out, where you simply just say, I don't know. And so now you have three potential outcomes. You have recovery, you have those who die despite maximal support, and then you have those where you say, gosh, I really don't know what your outcome was. I don't have enough information based on the observables to know what it could have been absent withdrawal. And as I'm describing this, those with a bit of statistical background in the audience may start to think about this like a survival analysis. Imagine a cancer trial where you give some chemotherapy and you look for outcome over time, but some patients are lost to follow-up. Those patients that are lost to follow-up fall into this category here, where you don't know what their eventual outcome would have been. So let's consider a simple experiment then, where you start with, say, 1,000 of our comatose survivors of cardiac arrest, and you input a whole vector of clinical data for each patient, EEG factors, imaging factors, their neurologic exam, their meds, et cetera. In total, about 60,000 predictors per patient. I mentioned to you that there's a lot of collinearity between these, and that's true. There's many correlations across EEG features. And so you need to use some sort of an approach to minimize that multicollinearity and get a stable signal. And one way that you can do that is a regularized regression or a penalized regression, which we think about often in terms of logistic regression, but has also been built out for survival analysis and for time series analyses. And in this sort of a regression model, you shrink the coefficients of strongly collinear predictors, potentially all the way down to 0. So you get some feature selection and you get some weighting. And without getting too much in the weeds, that process is guided through cross-validation. So you can iteratively train and then test the performance of a model to try to find the optimal fit. And the important bit here is this last bullet point, which is that when a patient is withdrawn as withdrawal of life-sustaining therapies, we censor those data. So we say we know nothing in this conservative estimate about what their outcome could have been. So the results of that, sort of very briefly, I think, without, again, getting too much into the weeds, we trained it on those 1,000 patients in Pittsburgh. And then we tested the data on about 250 patients from University of Alabama, another tertiary care center, who provides excellent post-arrest care, but may have very different practices about withdrawal of life-sustaining therapies. You get excellent performance overall. So this is a slightly bootstrapped sort of average performance here. Here we're predicting awakening. And so the sensitivity and specificity that's clinically interesting is up here, since we're predicting a positive outcome here. You get very good discrimination in the test sets, an area under the curve of about 0.93, which is incrementally better than some other models. And I think what merits highlight about the strength of EEG as a predictor is that of those 60,000 predictors that entered the model, it was almost exclusively EEG features which make it back out. So the 12 quantitative EEG features, which are primarily measures of amplitude suppression and frequency, contain virtually all of the information that is also contained in things like the CT imaging, in the medications, in the labs, in the severity of illness. And then motor exam also enters into the model. What I think is very interesting here is that in the external test set, the new cohort, this sort of a survival analysis makes substantially fewer errors in classifying patients in that external test set than a traditional logistic model, which I can't prove it to you, but I believe is sort of indirect evidence of the importance and the role of self-fulfilling prophecies in these analyses. So now I'm going to pivot a little bit to a conceptually different approach. So for those who are my age or older, you may remember this sort of life meter from early video games like Zelda. This is sort of the theme of conventional prognostication research. Find an individual patient and do some fancy modeling based on fancy predictors to come up with some sort of a life meter. And in this case, that life meter is a probabilistic estimate of their recovery potential, which can then be used to guide decision-making. Even with conservative survival models, there is the potential for self-fulfilling prophecies that are baked into these models, because even absent frank withdrawal, sincere and well-meaning ICU providers will titrate the intensity of their care based on their anticipated prognosis. And extremely attentive and more aggressive care may ultimately lead to better outcomes in difficult to measure ways that accrue over the duration of even a short critical illness. So what if we abandon this approach of trying to predict outcome and trying to predict recovery potential, and instead use this rich data set, which has signals of treatment responsiveness, has signals of secondary brain injury, to instead guide therapies and try to pivot more towards a precision medicine approach. This is some early work that we did, sort of just playing with that, a handful of quantitative EEG features, some CT imaging and exam. And we use some standard approaches for dimensional reduction to just try to cluster patients. And in this analysis, what you find is very clear separations of group that have clinically interpretable phenotypes. So very ictal or malignant EEGs are over here. Severe cerebral edema and an isoelectric EEG is over here. Folks who have a favorable exam and an okay CT are over here. And if you then take this one step further and say, well, what about the interaction between group membership here in this clustering analysis and outcome with consideration of treatment, you start to find some interesting things. So for example, there's an interaction here between your blood pressure target and group membership, such that patients in this group are more likely to do well when they have a higher mean arterial pressure. And that's something that we often modify in our care using vasopressors to try to drive augmented cerebral perfusion in the face of dysregulated cerebral autoregulation. Some of these patients don't benefit from temperature management at all. So it doesn't matter if you pick no temperature management, normothermia, 33 degrees, 36 degrees, they're too severely injured to benefit. building on this briefly is an ongoing R01 that I'm working with Karen Hirsch, a neurointensivist at Stanford, on that we cutely tried to name PreciseCap. And the hypothesis of this R01 is simply that not all post-arrest patients are identical, that this variability between patients can be quantified and can be used to guide precision medicine. So this is an observational study that's collecting high-resolution ICU waveform data, EEG, CT scan, clinical demographics, things like that. And it's embedded within a randomized controlled trial, which is an important concept here. So the parent randomized controlled trial is randomizing patients to different durations of hypothermia, irrespective of any clinical characteristics other than presenting a rest rhythm on which randomization is stratified. And so this provides, I won't call it a natural experiment because it's a randomized controlled trial, but an unnatural experiment that allows us to try to tease out an interaction between the care arm to which patients are randomized and their clinical phenotype. So these are a lot of complex data. I won't get into the weeds of the approach, but essentially we're taking all of the data that are available to clinicians, that are available to us, including the continuous waveforms from cardiopulmonary monitoring, EEG, brain imaging, meds, et cetera, and taking these multi-parametric, high-resolution, continuous or high-density data and using some sort of straightforward machine learning techniques like neural networks to try to find clusters of patients that represent phenotypes. And then after identifying phenotypes, we're then trying to use the phenotype based on these high-resolution data as a grouping variable and construct individual dose response curves at the level of the phenotype. And so, for example, there may be a phenotype who has cerebral edema, a flat EEG, refractory shock, for whom, in this case, the outcome 90-day modified Rankin scale is always poor. Doesn't matter the duration of hypothermia too, these are treatment non-responders. And conversely, there may be a phenotype of patients with relatively light brain injury quantified by any of the modalities that I've described thus far, who has both non-zero outcomes, but also who have some optimal duration of hypothermia. And this analytical approach, the idea of fitting a piecewise model to find this inflection point here, which is the shortest duration of hypothermia that maximizes outcome, is mimicked or built on the overall analytical approach for the parent R01. So I don't have results of this to share. We're just trying to spin up our data collection, hopefully enrolling our first patient in the next couple of weeks. But I think this sort of highlights what I am hopeful will be the future of prognostication research after cardiac arrest, not just in the case of EEG, but more broadly, which is instead of trying to predict ultimate patient outcome in a fraught scenario that is hopelessly confounded by withdrawal of life-sustaining therapies, we start trying to make a more clinically useful prediction, which is for the patient I'm treating at the bedside right now, what is the care that they need that will maximize their potential for recovery? And with that, I will stop and thank you again for your attention and the opportunity to speak with this audience.
Jonathan, thanks. If folks have questions, go ahead and put them in the Q&A, and we'll try to pass those along. I think I'll begin with Dr. Bahir. How do you account for the treating team bias you mentioned? So what is the story on misspecified outcomes?
Yeah, that's an insightful and a hard question. I think that broadly speaking, there's two approaches when you're worried about something like this. One is to try to find ways to quantify that factor, which currently we would consider as an unmeasured confounder. So if you can measure it, then you can adjust for it statistically. And one thing we've tried to do is to look at our historical practice patterns, line it up with things like the call schedule for who is on call, and then try to disentangle, like, what is the provider effect, and what potential biases is introduced by the individual provider as a proxy for measuring that intangible. And that hasn't worked very well because it turns out it's not just the provider on call who probably influences these things. It's a complex dynamic of team and nurses and family and all these other things. So I think absent being able to measure it, then you have to do one of these things that actually does not rely on understanding the effective intensity of care or the treating team. So you can, again, sort of censor at withdrawal, or you can try to predict something that you think is less likely to be biased by the treating team. That's, I think, likely to be more fruitful going forward than trying to find proxies to measure those intangibles.
Couple more questions. One is, can you use changes of EEG over time as an alternative or surrogate outcome measure independent of withdrawal of life support?
Yeah, so no, I think probably not. So this gets down to the problem with informative missingness and the fact that the data are censored not at random, but also to the generalizability. So I didn't say this explicitly, but one proposed solution is that if you simply made your prediction based on the cohort of patients who did not have withdrawal, which is, I think, another way of saying EEG over time, because you can't have EEG over time if there's no withdrawal, that you might be able to protect yourself against those self-fulfilling prophecies just by making predictions on those who have a ground truth outcome observed. The problem with that is that those patients are systematically different. And so you end up with a model that just has no support for prediction in the feature space where withdrawal of care happened. Those patients are fundamentally different than those who had a ground truth outcome observed. And so you're asking the model to make predictions in a cohort where it's never had the opportunity to learn, and it performs poorly.
Pat, you have a question if you want to.
I do. Jonathan, I really enjoyed your talk. I think the idea of bringing together the camps, whether you're in the EEG camp or the imaging camp or the SSEP camp or the biomarker camp, and bringing them together is really key. And but what I'm wondering is the electronic health record rich enough on the topic of limitation of care? Do we need a much more accurate and sophisticated input on that? For instance, in pediatric critical care, you might have withdrawal of care or add no new support or different levels of that. And is that really captured adequately?
No, it is not captured adequately. So for our analyses, we use prospective registry that's maintained independent of the EHR. Where we code the proximate cause of death. And we follow the convention that was started by the Resuscitation Outcomes Consortium for some of their data collection, where we ask the post-arrest service provider who was involved in those discussions whether the death was due to brain death, was due to withdrawal of life-sustaining therapies for perceived poor prognosis, withdrawal based on the patient's values and preferences, or multi-system organ failure. So that, I think, is a step up from what the EHR does, but is probably still inadequate because it treats those outcomes as independent and mutually exclusive, when in reality, we know that patients often have bad multi-system organ failure and brain injury and the decision making is more subtle and complex to simply saying it was A or it was B. So I think EHR definitely not. A prospective registry is probably a step better, but may not be sufficient ultimately to capture the nuance of those complex decisions.
Jonathan, I'm going to bundle two questions and then wrap us up so we can try to get back on schedule. And thanks for moving your talk up to help us. The first one is: has there been consideration of using AI like DeepMind or other large artificial intelligence approaches that have been advertised heavily to find patterns in the data that predict outcome? And the other one with that from Dr. Xu is any EEG features of near-death experiences or heightened consciousness near death and recovery?
Okay, great questions. To the first, yes, there are many groups, including our own, who are using various AI approaches to try to make these predictions. My personal bias is that may not be the answer here, in part because no matter how sophisticated your AI is, if they're learning on the observed outcome label, there's still these potential for self-fulfilling prophecies. And in part, it, depending on your approach, may come at the expense of interpretability. And I think for life or death decision-making in the ICU, physicians and families will demand some degree of interpretability. I think AI may have a valuable role in trying to elucidate some mechanistic hypotheses that could then inform future modeling and how we choose to featurize or consider these EEG, and that may be a more useful tool for it. The second answer, or the second question, which was about near-death experiences, I truly don't know. That's outside of the world I live in, and so I can't give you an intelligent answer there. I apologize.
All right. Thank you so much.
Thank you.